The Indian Institute of Chemical Biology. Much more than just a name to me. Much more than an institution for scientific research. It was perhaps my destiny that I'm here today for an interview as a research student.
Ms. Banerjee, we have seen your papers and you certainly qualify, but we are not here just to look at the papers, but also to understand the person.
Sir?
We want to know to whom we are admitting to our research fraternity. May I know why you have chosen to apply to IICB?
So you must have seen my papers which show my areas of interest as well as my background.
Yeah, we already have, yes.
But why IICB? You could have applied to other institutions, even universities abroad.
In fact, we would like to know whether you have applied elsewhere.
No, sir, I have not.
And why not?
My reasons are personal.
Could you please elaborate?
Certainly, sir. You may not remember I was sent here by my father, Professor Sitanshu Banerjee.
Oh yes, I know him as a very respected teacher of applied physics.
So you may then also remember that my father had sent me here for my DNA test. My elder brother is suffering from glaucoma.
E. D. N. G. 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 K. G. 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 K. L. A. E. M. M. C. U. No point in getting upset, Ma. We always knew this would happen to me.
However much I know, it's not easy for a mother to accept her children losing their...
I haven't lost my eyesight yet, Ma. Not yet.
Please, Ma. Please don't cry.
It's only a child.
Legacy, Ma. It's in our blood. Who can we blame?
We had taken my brother from one specialist to another, but the answers would always be the same. Glaucoma is a slow but inevitable process. His eyesight would progressively fail, and the first major indication was tunnel vision. My brother's vision would gradually be reduced to a tunnel, till darkness overcame his world, total darkness.
First, it was your father. Now it's your brother. How I worry about you.
I'm quite okay, you know.
But for how long? Can you tell me that?
For the rest of my life, I suppose.
Is there any point in going on with your studies? Should you even try for further studies? I wonder, what will you gain?
Manushi, I've told you, learning is not about gain. It is an experience, complete in itself.
Experience? Where will that experience take her in life?
But Ma, Why give up my studies even before the first symptoms appear? You don't even know if I'm also affected.
A lot's happening in the sciences, Manushi. They can find out a lot of things today that wasn't possible in my time.
It wasn't possible for your son either, was it?
No, it wasn't. But Rupa may be the lucky one. There are genetic studies now, DNA. That might be able to help her.
Don't do this to me again. Don't give me hope, which I know will come to nothing.
It's true, Manushi.
What Baba says is right.
And Baba is always right. He knows. But your Ma, she just has hope. She must keep faith. You don't know what it means to hope against hope for your children, and then they're destroyed.
Rupa, why don't you go to IICB,
What's that?
the Indian Institute of Chemical Biology? They're working on the Human Genome Project. They might be able to tell you if you carry the genetic strain for glaucoma. I know Siddhartha Rai, Professor Siddhartha Rai. He's the director of IICB.
That's how I came to IICB first, to the Human Genome Project of Dr. Kunal Rai.
Rupa, what I was been telling you is that in case of juvenile and some form of the adult onset glaucoma, the change in the DNA can actually cause the disease. And that can then whenever there is a change in the DNA, then the disease can run in the family, and sometime it can be very predictive. Okay, and so one has to really track the defect in that case.
What do you mean by defect?
Well, that's an interesting question. You see, DNA is actually a polymer of 4 different chemicals, and you see represented by 4 letters here. And when there is a change in the sequence in any of the letter, that actually where the defect might occur, and it could cause the disease.
So how do you really find out?
Okay, so that I'll tell you briefly. So essentially what you do is that basically we will take the entire DNA that we have, and we'll go and specifically amplify a segment of the DNA we call gene of our interest. Well, to make it simple, you are reading a book and you are very interested in one specific chapter. So you went, you copied that particular chapter, and maybe your friends are interested, quite a few copies of that particular chapter, right? So here what we do, that in the entire genome, our target sequence, we copy it many times. That's what you call is amplification. Once you did that, and here what you do, then you can see this DNA for specific region you have amplified, it will come like bands in the gel as you see in this picture. So I guess you guys are working on that now, right? So you understand that for doing this kind of PCR, it's very important that you have a negative and positive control. Could you tell me, Deblina, that why you need a negative control?
It doesn't contain any DNA, and so there is no amplification.
And why you think we need a positive control as well?
To make sure the amplification is specific.
Very good. So you see, even though, Rupa, if you notice, even though we have patients and the normal individual samples, they look exactly the same. So the question is that how is it that you find out that there is any defect, right? So for that, once you do the analysis, and this we have done already on one sample, we are showing the example. You see here, actually these are 2 chromatograms. One is for a normal individual and one for the patient in the same family. And if you follow, you find out that in both cases the sequence of letters are exactly the same, excepting in one case, in the patient you see there is an extra peak, and then there is a different letter from C to T.
But isn't it possible that different people might have different DNAs anyway?
See, you are already in the game. Absolutely. What it is that you have to take this sample from the family and analyze in the family itself. Just the reason we ask you all these questions. So here, if you notice in this family that all the individuals who have the defect also have the disease, and all the individuals who are normal don't have that particular defect in the DNA. None of the individual who are not affected here in the first generation and the second generation, you see that they do not contain the T. Therefore, those individuals who do not yet have reached the age for the disease, there when the gene is analyzed, one would know from there that where they contain the defect, and therefore make a prediction that whether the person will come down with the defect. So in case of your family, we'll go follow exactly the same steps and we'll know the result.
So when do I get my report?
Well, it's going to take a few days, of course, but you seem to be pretty worried. Actually, there is only 50% chance that you might have the defect, but 50% chance that you wouldn't have it, right? Let's keep our fingers crossed and see what happens.
That was the moment, I suspect, when IICB and my destiny became inextricably linked. Sharing a cup of coffee in the canteen with people I had just met seemed like meeting long-lost friends.
They've constructed it out of those nucleotides.
IICB seems like a large organization. How many scientists work here?
About 85 of them, and up to 50 researchers, and another 200 odd technical and support staff.
All working on the Human Genome Project?
Oh no, work is going under 6 major divisions.
6 major divisions?
Yep.
You look too young to be a PhD student.
Well, yes, I'm a student of plant chemistry.
Plant chemistry?
Yes, I analyze plant extracts and I see the medicinal properties of their compounds.
Such as?
Such as a very useful little anti-cancerous compound extracted from the betel leaf. Would you like to meet some of my professors?
Sure.
Come on then.
We have isolated a compound called chlorogenic acid from piper betel leaves. Actually, this compound is very effective in destroying a special type of human cancer called leukemia. We have tested this molecule in animal model called nude mice model, where same type of cancer cells have been grafted, and it appears to be effective on the human cancer cell type in the animal model.
So it may be used as a drug to cure cancer in future?
The answer is not so simple, because if you take a look on the history, out of 100 molecules discovered in the lab, maybe 10 or less than 10 appears drug. So only time will tell.
So why didn't you tell Rupa about the new things that we've discovered, new drugs discovered in the lab?
Recently our institute have developed a new drug. Technology has been transferred to the industry, and that technology they are marketing the prostatic problems drug in the name of Prostaline.
It was a fascinating world that opened out for me. Not that I even began to understand even a fraction of it, but I got a sense of the tremendous energy and the purpose that was inspiring people at IICB. That touched me very deeply. I wanted to be a part of it.
You know, scientists here works with so many compounds, say around 10,000 molecules, but it may take them a lifetime to find out the actual molecule they are looking for. But who knows, they may find it out tomorrow itself.
Thanks. I think your work here truly touches our lives. It might certainly touch yours.
We just hope that Dr. Kunal's DNA test will help you.
There must be a lot to explore around here.
Oh yes. If you like, I can show you around.
That's Dr. J.C. Roy. He founded IICB as far back as 1935.
As early as that?
Yes. He was a kind of crusader. He fought against Kala Azar and malaria. He fought against all sorts of things, really.
Like what?
Like superstition, like ignorance, like epidemics and diseases that plagued rural India at the time.
Did he make a difference?
Oh yes. By the late 1930s, IICB had a profound influence, and Rabindranath Tagore wrote about the glowing work that he had done on an India that was still a colony. Professor Bachawat transformed the institute into an organization of all India importance. The term chemical biology was coined by him.
I must find out more about the history of IICB.
Yeah, you should. It's very inspiring. But I think you'd like to focus a little bit more on the present and not just the past.
Like what?
Well, like the resurgence of interest in chemical biology.
What's that?
It's a scientific discipline that combines both chemistry and biology. What happens is it uses chemical tools and techniques as well as utilizes life sciences by biological compounds. Chemical biology tries to understand and manipulate molecules of a living system. If you understand the chemical aspects of a disease, then people have a greater chance of finding its cure or prevention.
You mean people like me?
Well, I wouldn't know that, but Dr. Adya might.
What's Dr. Adya's field?
We'll soon find out.
Dr. Adya, could you tell us a little about your current research in gene therapy?
Many complex human disorders, as well as aging, are associated with dysfunctions in the main energy generating systems of the organism. Now, at the center of the energy generation are the mitochondria, which use the energy of foodstuffs to convert into adenosine triphosphate or ATP, which is the universal energy currency of all cells.
Interesting. Could you tell us a little bit more about these mitochondria?
Well, mitochondria are small compartments within all cells which have their own genetic material or DNA. Mutations or other changes in this genetic material can lead to faulty ATP generation and therefore to disease.
So what is IICB doing about it?
Well, here at IICB there is a large group of scientists working on a major supra-institutional project aimed at looking at mitochondrial dysfunction in disease and also trying to figure out new approaches to correct these mitochondrial deficiencies.
And how do you propose to do this?
Well, earlier we had this observation that the protozoal parasite Leishmania, which is a single cell organism, has this miraculous capability of being able to transport RNA into its own mitochondria. And then we made the exciting observation that a complex of proteins from this parasite can be taken up into human cells.
This is published in the top international journal Science and is widely cited and is written by Dr. Adya himself.
Right, and what we hope to do as a result of all this is to use this complex as a vector to deliver correctional ribonucleic acids into mitochondria and thus correct the dysfunction due to disease.
So then we can expect very advanced mitochondrial therapy from IICB in the future.
Certainly you can.
At home, I was a silent witness to what genetics was in a way that I do not wish anyone else to see. The pain, the agony, the sharing.
Baba?
Let Mozart speak.
Every day I returned to IICB, there were new experiences together, new horizons that opened out for me. It was nothing like anything else that I had ever seen. It was far more than discovering the frontiers of science. It was more like discovering myself. That's where I saw you for the first time and met you as well. You spoke for a long time about the power of fundamental scientific research, without which science can have no application. I remember every word, sir. You spoke about my father as well.
Your father was an inspiring teacher. He encouraged his students to become excellent. And this is the excellence that we also pursue at IICB.
That will mean a lot to him, sir, when I tell him that.
It is unfortunate that today not many find teaching profession and teachers to be inspiring, but he was one of the rare ones who did so.
Students still come to the house, in reduced numbers, of course.
Yes, I understand. Science is changing every day, and it's such a huge challenge that you need dedicated people like you to make the difference, to meet the challenge.
Of course.
What about the test that you came for?
They're under processing, sir.
I wish you all the best, because the future belongs to people like you.
I hope I can live up to your expectations, sir. In these few days, I've learned a lot about IICB's wide range of research and publications.
That's right. In the past, we have earned worldwide recognition on leishmania research, on cholera research, and discovering drugs from natural resources, that is plants. In the future, we would like to apply modern technology like proteins and proteomics to solve the health problems of the country like asthma, cancer. In fact, we want to develop a whole new campus in the Salt Lake devoted to proteins and proteomic research only.
Yes, sir, I know of IICB's 6 major divisions and its common goal for the understanding molecular basis for drugs, vaccines and diseases.
Sir? May I come in, sir?
Yes, please. Take your seat.